Hello everyone, myself Ishita Sharma, and in this video I am going to talk about erythropoiesis or the formation of RBCs. So in previous video we discussed about various characteristics of an red blood cell. In this video let's just first talk about structure of RBCs. So here in this picture you can see the structure of an erythrocyte whose main function is to carry oxygen throughout the body. So a human RBCs is bound by a elastic semi-permeable membrane, as you can see in this picture, which enables it to squeeze out through narrow capillaries. The outer surface of cell membrane has a protein called glycophorin. So this glycophorin contains the blood group antigens, and the inner surface has a this surface this surface has a contractile protein that is known as spectrin, and this spectrin protein provides the biconcave shape to the RBCs. As we discussed in previous video that RBCs lack nucleus and mitochondria and various cell organelles. The main feature of an mammalian RBCs is characterized by the absence of nucleus, mitochondria, ribosome, centrioles, endoplasmic reticulum, and many more other cell organelles. Why this happens? Because these RBCs, their function is to carry oxygen and to ensure more and more transport of the oxygen throughout the body. So the that's why these organelles are not used here, because it may take a lot of space, and uh, there will be the low consumption of oxygen if these organelles are present, right? So the RBCs are primarily concerned with the transport of oxygen from respiratory organs to the body tissues. Now let's just discuss about the molecular structure of an hemoglobin. So hemoglobin is a purple colored iron containing respiratory pigment that is present in the cytoplasm of RBCs, right? So hemoglobin is a conjugated protein that is formed of four heme groups. Here we can see the heme group, and there are four among them. Each heme group contains an iron containing porphyrin ring. Here we can see this. Formed of these porphyrin rings are formed of four pyrrole rings. So another thing is globulin. So globulin is formed of four polypeptide chains, out of which two alpha chains are there and two beta chains are there. You can see the beta chains and alpha chains in this structure of hemoglobin. So each alpha chain is formed of 144 amino acids, while each beta chain is formed of 146 amino acids. Here we can see alpha chain, this is the beta chain. So one RBC has about 280 million hemoglobin molecules. So an RBC is considered as a living bag of that contain hemoglobin. So about 95% of dry weight of RBCs is due to hemoglobin, because this hemoglobin is responsible for the transportation of oxygen, because it has high affinity for oxygen. Now let's just discuss about the quantity of hemoglobin in the human body. So the amount of hemoglobin per 100 ml of blood is called hemoglobin count, and it is measured by an instrument called hemocytometer, as we discussed in the previous video. So the hemoglobin content that is present in female is about 12 plus 2 gram percent, whereas in adult male it is around 14.5 or 13 to 15.5 gram per deciliter. And the hemoglobin count is highest in the fetus, because it needs more oxygen, that is 23 gram per 100 ml at birth. Now let's discuss about the lifespan of RBCs. So RBCs, that there is no nucleus, so that's why RBCs are short lived. They don't live up to long time. So old and worn out erythrocytes, these erythrocytes are destroyed in the reticuloendothelial cells of liver, spleen, and bone marrow, and this process is called hemolysis. In this picture, we can see the hemolysis, that is degradation of RBCs in spleen, liver and bone. So the hemoglobin of hemolyzed erythrocyte is changed into another iron containing pigment, that is hemosiderin, and the bile pigment bilirubin and biliverdin are formed from it. So when these RBCs are degraded or hemolyzed, this iron is stored in the liver as ferritin, and it is again used in the formation of new hemoglobin, and while the bile pigment bilirubin and biliverdin are expelled out of the body along with the fecal matter. So each RBCs take about 50,000 circles or cycles in the body before hemolysis. So it transfers a lot of oxygen, right? So RBCs are hemolyzed at a rate of about 2.5 million per second. So 2.5 million RBCs are destroyed per second and also formed at the same rate, so their number is maintained in the blood. So now let's just move to the core topic of this video, that is erythropoiesis. So the formation of RBCs is known as erythropoiesis. So the organs forming red blood cells are called erythropoietic organs, right? So this erythropoiesis occurs in the presence of a glycoproteinaceous hormone that is known as erythropoietin. So these erythropoietin are formed by the kidneys. This kidney forms the erythropoietin hormone. So in human being, the entire process of erythropoiesis is completed within just 72 hours. And in the developing fetus, the hematopoietic tissues are present in the spleen and the liver. And when the person grows elder, these RBCs are mainly produced in the red bone marrow and in the skull, ribs, sternum and vertebrae in the adults. So as we know, the erythropoiesis occurs inside the bone marrow. So volume of bone marrow at the time of birth is 70 ml, which increases to about 2000 ml in the child and 4000 ml in the adult, right? So erythropoiesis, if we talk about erythropoiesis, it is a self-regulatory process, right? And it is controlled by a feedback mechanism which depends upon the oxygen requirement of the tissues. So when the tissues require more oxygen, more and more erythropoiesis occurs, and more and more erythropoietin hormone are produced by the kidneys. And so this hematopoiesis is controlled by iron, right? Because heme is iron and proteins, while the maturation of RBCs is controlled by folic acid and vitamin B12, that is called erythrocyte maturing factor. So vitamin B12 and folic acids are known as erythrocyte maturing factor. So therefore, deficiency of this vitamin B12 and folic acid may cause anemia, right? So deficiency of vitamin B12 called pernicious or megaloblastic anemia, as we discussed in the previous video. So the excess of erythrocytes that are present inside the blood is stored in the spleen, which act as a blood bank of the body. So spleen also act as a graveyard of RBCs because the hemolysis of RBCs takes place here. So guys, that was all about the erythropoiesis or some lifespan of RBCs. I hope you like this video, and if you like this video, don't forget to share this video and subscribe our channel. I will meet you in the next video as soon as possible. Till then, take care. Goodbye.